Hello, everyone. What we're going to do in this lecture, we're going to go through a quick, and I stress quick, overview of the cellular structure. So what we'll talk about, first of all, is how did we get to know that all living organisms are made of cells? Which scientists contributed to the discovery, ultimately, of the cell and come out with that theory, unifying theory that's referred to as the cell theory. Second thing we'll talk about is the features of cells. We'll start by talking about the common properties that all cells, whether we're talking about a bacterium, a eukaryotes, a plant, these features, the structures, they're shared by all cell types. And then we'll get into the specific of the different cell types. So we'll talk first about prokaryotic cells, then eukaryotic cells, and finally, we finish talking about a few structures that are unique to plant cells. So historically speaking, the first time the cells were described were in the mid-1600s, so a long time ago, by this scientist named Robert Hooke, that he was actually playing around using this very first type of microscopes, light microscopes, similar in some way to the microscopes that we normally have in the lab, our labs, obviously much more rudimental ones. And he was looking at different structure, and what he had noticed, and this is one of his drawings, by the way, that he published, what he had noticed is that if you look at cork, slices of cork, so from different plants, you would see, from trees, you would see that there were these tiny little repeated holes, right? You can see here in this drawing right here. And so these will be present in all different corks that you would be looking at. Because of the similarity to what, to the monk cells, you know, monks are these sort of priests, religious figures that used to live in a rough environment, tiny holes on the walls, tiny holes like this one. So because of that, it called them like cellulae, similar to what these monk cells were called in those days. So hence the name cell, or cellula. It will be in Italian, cell in English. Despite describing for the first time the existence of cells, I want to stress on one thing, is that Robert Hooke did not understand what they were about. He just noticed this hole, first person to describe them. Now, the first association between cells and life is attributed to this other scientist, Anton van Leeuwenhoek, in the late 1600s, so a few decades later. And he was looking at different samples, from ponds to biological samples and so on. And what he was noticed that in this sample, he could actually see these tiny little things similar to cells that could actually moving. So they look like tiny little animals, animalcules, or whatever you want to say. Sorry, the pronunciation in English. And so this is, for the first time, he actually described the protist, sperm cells, bacteria. Was the first time that these organisms are described. These are some of his drawings here. And these also was the first time that was showing that there was life, that it was basically invisible to our naked eye. Now, later on also, this was demonstrating that, so the association between the cell and the reproduction and so on comes from these two scientists. These were both botanists. First one is Robert Brown in 1820s, that for the first time actually he observed, looking at different plant cells, he observed that they had this central structure. In Latin, central is, say, nucleus, and hence the name nucleus, or associated with this structure that we still use nowadays. Together with that, still in the first half of 1800, Matthias Schleiden, by the way, Robert Brown is also that same scientist that is well known for the Brownian motion theory. Matthias Schleiden also was the person that noticed how these nuclei were present basically in several different types of plants. And so he theorized that the cell and the nuclei are indeed essential components, essential building blocks of plants. Not only that, but he was the first person that theorized that the nucleus, as the cell, is important for the development of the organism, and the nucleus is important for the development of the cell itself. Finally, last scientist we talk about, you don't need to remember, by the way, all of these names. It's just to give you an historical perspective of how we got to know about the cell and its role in living organism. So Rudolf Virchow, so now we're talking about mid-1800s, he was actually the first person that described one very important thing, that cells can actually divide. Cells are actually living organisms, seem to be living, tiny living organisms. First time they described that, this was actually destroying the common theory in those days. You have to understand that in those days, they used to think that life would arise out of nowhere. They were used to see, for example, water becoming cloudy, in ponds, or seeing organisms coming out of the pond. But what they thought it was that they were coming actually forming out of nowhere. So now this discovery showing that cells can actually divide was actually pointed to a fact that you can actually have to these even invisible creature actually always arrive from previous living organism, previous cells. So there is life associated with these cells, and that's the base of the formation of new organism all the time. So combining all together, scientists finally came out with what we refer to as the cell theory, which is basically one of those very important points or properties that we discussed at the beginning of the course. Properties of all living organisms is that all living organisms are made by cells. That's exactly the cell theory. That's what it talks about. So the cell itself is the basic structural and functional building blocks or unit of all living organisms. All life exists with cells in it. There is no such a thing as a living organism that is not made by cells. Viruses, again, I stress, are not living organisms. And then not only that all living organisms are cells, but also that new cells can arise only from previously existing cells. So this is the cell theory that all these scientists combining together, basically, their evidence, their observation came out with. Now, moving on to cell structure. Before we talk about the difference between prokaryotes and eukaryotes, then we'll talk about plants as well, which are part of eukaryotes, by the way. Now, just a few words about the division of all living organisms on Earth. They are divided into these three domains. We have bacteria, archaea, and eukarya. So bacteria and archaea used to be grouped together as prokaryotes, and we still talk about prokaryotes. The reason they were grouped together is because of morphological similarities. They look very similar, they share different properties. However, evolutionarily, they're not really related. Okay, so now they're separated within as bacteria, archaea. However, in the rest of the lecture, I will refer to both of them. We talk about property of cells as prokaryotes, okay, using the old definition. And then we'll talk about eukaryotes, which are obviously very different than bacteria and archaea. So starting from common properties of all living organisms. First thing is that all living organisms, they're made of cells which cannot grow indefinitely. So, and that's the reason why cells are always small units. Think about it. Cells could have grown theoretically. You can imagine, why don't we have cells that are 10 centimeters or 4 inches long? Why don't we have them? There are some, but they're made of fusion of different cells, not single cells. So all cells are very small units. Like, look at bacteria, they're all in the range of a few fraction of micrometers to 5 micrometers. Eukaryotic cells tend to be about 10 to 100 times larger than bacteria. And this size is limited. In neither groups, the cells can go any further than 5 or 10 micrometer. Why? There is a reason. The reason is that if you think, when a cell is growing, all the metabolism inside the cells, and this includes both the reaction that builds up the things, so, and the requirement for energy, and the requirement for building blocks like amino acids and so on, as well as the production of waste, will grow proportionally to the volume that the cell is growing. However, if a cell is growing, obviously the membrane is growing too, but the membrane is growing slower. And the membrane is essential to move all these building blocks, amino acids, and waste in and out. It's necessary. So you have, if a cell keeps growing, you have the growth of two things, of metabolic needs and need to move this material in and out. The problem is that the metabolic needs grow proportionally to the volume, which grows proportionally to the power of 3, all right? Whereas the surface grows proportionally to the power of 2. I'll give you an example. Imagine this being a cells. Cells are not really square or cuboid like these ones. So imagine this is the cell that keeps growing. And here you can see how this cell will be growing over time, or the surface and the volume will be growing. You can see that if you can start from a point where the volume is actually smaller overall as a value than the surface area. And then the more it grows, the volume keeps growing faster than the surface, to a point that actually, numerically speaking, is much bigger than the surface itself. So if you associate the metabolic need to the volume and the capacity of moving material in and out to the surface area, you can see how the metabolic need is growing so fast at the power of 3, whereas the capacity of sustaining it, bringing building blocks in and removing waste, grows lower than that, grows at the power of 2. So for this reason, cells can keep growing only up to a certain point, which basically corresponds to a certain ratio of the surface area to the volume. So each cell would have its own surface area to volume ratio maximum value there. What happens if a cell is going past this? There are different options there. The cell can stop growing and become quiescent. So basically, it will slow down or stop the metabolism. The cell can die, or the cell can divide. And when it divides, obviously this ratio will go down to a lower value, and then this cycle can last. Now, this number here, the ratio between surface to volume ratio, is proportionate to the metabolic activity of a cell. So if a cell has a lot of metabolism going on, constantly producing protein and so on, it would tend to be smaller. Whereas if a cell overall, let's say, is a structural cell, you can see some plants, some very elongated cells that their function is just structural. They can grow longer because at this point, the metabolism is still not growing as fast as it would be in a very active metabolically active cell. Now, what are the common characteristics of all cell type? Now, whether you're talking about a prokaryotes or an eukaryotic cell, there are 3 common properties. Here you see only 2, but there's a third one in the next slide. There are 3 common properties that all cells have. They all have a plasma membrane. This is made by a phospholipid bilayer with proteins, and which basically creates this barrier between the external environment and the internal environment. Now, the internal environment in all cells contain this liquid fraction, which is referred to as the cytosol. This is the aqueous solution, which has the ions and all the organic molecules, protein, carbohydrates, and so on, which are within the plasma membrane. So if this is the plasma membrane, this is the external side. Everything within this plasma membrane, so on this side here, will be or the liquid component will be referred to as the cytosol. The third component that all cells have is a region which is dedicated to the protection and the execution of all the information stored in DNA. Now, when it comes to prokaryotes, this region is non-membrane bound. There is no real nucleus. There's no real nucleus. In fact, prokaryotes means before the nucleus. And still, despite that, it's a very clear, distinct region from the rest of the cytoplasm. You can see it here in this picture of this bacterium. This is the nucleoid. You can see it just from the color. This is an electron microscope picture. You can see how different is the contrast. So it's a very different region. It has the DNA, but not only there, the circular molecule of DNA, but it has also all the proteins, so DNA polymers, all these proteins, enzymes that are necessary to maintain, and execute the information stored in DNA. In eukaryotes, instead, the region is much more clearly separated from the cytoplasm through the presence of a double membrane. We'll talk about that in the next slides. So differences now. In prokaryote cells, the prokaryote cells, first of all, they're all smaller compared to eukaryotic cells. We talked about that earlier on. They're about 10 to 100 times smaller than eukaryotic cells. They can have different shapes, three main types. So you can have rods, like this bacillus here. You can have spherical bacteria, which can exist as single unit, as pair, or chain of the spherical unit, like in this streptococcus. And then you can have spiral bacterial as well. Together with that, all prokaryote cells, they always have a cell wall. Okay, this is a cell wall that provides strength, if you want, to the cell membrane. In fact, as soon as you break down the cell wall, the cells tend to explode if you do that in a lab. And so it's made by proteoglycans, which are basically carbohydrates joined to proteins, and so they provide rigidity to the cell itself. Together with that, bacteria also have an additional feature outside the cell wall, which is referred to as the capsule or glycocalyx. So you can remember capsule, it's easier to remember. And this still has a function of protecting the cell, helping the cell to adhere, to attach to different surfaces, protecting also from water loss and physical damage in general. Now, other unique features of prokaryotic cells, whether you're talking about an archaea or a bacterium, they do not have organelles, so there is no functional region within the cytoplasm separated by membranes. However, some bacteria, like the cyanobacteria, those original, very old bacteria, you remember those bacteria that are thought to be the predecessors of chloroplasts. So these bacteria do have invagination of the external membrane. So this is a picture of a cyanobacteria. So what you have, you can see here some structures. So you have that the cell membrane actually can form folds inside, organized like this, which is where the pigments involved in photosynthesis are concentrated. So they form this structure, which are similar to the thylakoids, if you want, in the chloroplast, which have the function of capturing the energy, the photons coming from the sun. Together with that, they have a cytoskeleton. Eukaryotic cells have a cytoskeleton too, but the structure is different. It's made by filaments, in this case, now but 3 different types. And also, similar to eukaryotes, they have flagella for the movement. However, flagella are longer structure than what you see in here. However, they're made by different proteins, referred to as flagellin, whereas in eukaryotes, you will see the flagella are made by microtubules. But these are units that are used for movement. Even the movement is generated in a different way in prokaryote cells compared to eukaryotes. Moving on to eukaryotic cells. Well, you perfectly know eukaryotic cells have different properties compared to prokaryotic cells. Eukaryotic cells, first of all, they're well known to have organelles. So they have these membrane systems, these structures inside, which have specific function, which are referred to as organelles. Okay, they can be interconnected, attached to each other, or separate. They may have different function in general. They have also a cytoskeleton. Prokaryotes have it too. However, in eukaryotic cells is much more complicated. It's formed by 3 different structures there. You have intermediate filaments, microtubules, and so on. And then you have, I don't have a picture, sorry, I thought I had it. So it's formed by different components and has also different functions. Together with giving strength, obviously, to the cell, it allows also the cell to change shape. Imagine like an amoeba, for example, moving around. It moves around because the cytoskeleton allows to do that. It changes shape to the cell. Also, it's used to support organelles, which are not present, obviously, in prokaryotes. and also helps, you see, when we talk about the cell division, it will help also with the separation of the chromosome during mitosis and meiosis as well. Now, when it comes to cell wall, not all eukaryotic cells have a cell wall. We don't have, for example, our own cells do not have a cell wall. However, it's present in some eukaryotes, such as fungi, plants, and several protists. Together with that, eukaryotes, as you know, they have a real region or true nucleus, so a region which is membrane bound or double membrane bound, which is dedicated to protect the DNA and contains also all the proteins related to its function and storage. Moving on to the organelles. So that's a very unique feature of all eukaryotic cells. All eukaryotic cells, they have organelles, which can or might not be connected functionally and structurally. Now, there is a group of organelles and structures which are connected with each other, which form what is called the endomembrane system. This is formed by different organelles. We'll see them in the next slide, which do share or somehow they are continuously connected in terms of function. You see, for example, the endoplasmic reticulum, it's involved in the modification of proteins, and then it ships this protein to the Golgi. So they are connected both structurally through vesicles and in terms of function as well. And they have the most diverse functions. So we have modification of proteins, you have synthesis of lipid, detoxification, regulation of ions, particularly of calcium concentration, and the proteins, a transfer of also proteins from the inside to the outside as well. If you think of through the formation of vesicles and also the digestion of any material that's brought in. So there are several functions which are shared by all these components in the endomembrane system. Some of them are unique of some of these compartments. So what are these organelles? And you see well in this picture. So the first part of the endomembrane system is actually the nuclear membrane itself. It's a double membrane, and so inside it actually has its own lumen, which is in continuity with the rough endoplasmic reticulum. You can see it here in this picture. It's connected through this little channel here, membrane channel. And so the first part is the nuclear envelope. The second part is the endoplasmic reticulum, both the rough and the smooth endoplasmic reticulum, which then connects through vesicle to the third component, which is the Golgi. We'll talk about all these organelles in the next few slides. And then these is connected with these vesicles that are produced, like lysosomes, which are digestive vesicles, or vesicles that have the function to ship the material to different organelles or to the outside through the fusion with the last component, which is the plasma membrane, last component of the endomembrane system. So all of these organelles and structures, such as the plasma membrane, are part of the endomembrane system. Now, starting from the first component, the eukaryotic nucleus. So this is that region, again, I'll say it over and over, that has the function of storing the DNA, protecting it. Not only that, but also making it functional, execute the information there. So that's where you'll see when we talk about transcription, that's where it takes place. And then the RNAs move from there into the cytoplasm where translation takes place. Now, it's formed by two membranes which have a lumen inside, which is in continuity with the rough endoplasmic reticulum. In fact, if you look at the structure of the double membrane, you see that it looks like what you see later on, like the rough endoplasmic reticulum. And in fact, it has a ribosome attached to it. Now, these barriers, this double membrane represent a big barrier between the cytoplasm and the inner part of the nucleus. And so the movement of material between these two compartments in one direction or the other is strictly regulated through these structures made by hundreds of proteins called nucleoporins. These structures are called nucleopore complexes. Each one of them is one complex, which basically tightly regulate what moves in and out. Just to give you an idea, proteins that move, they're synthesized in the cytoplasm, but they have to move inside the nucleus they need to have a specific sequence of amino acid called the nuclear localizing sequence, or NLS, in order to be bound by the nuclear pore complex and be transferred inside. Same thing for the mRNA, moving from the inside to the outside has to have specific function, structure, in order to move from one compartment to the other one. So they tightly control the movement of material from one side to the other one. Now, inside the nucleus, there is the DNA, is in the form, when the cell is not dividing, is in the form of chromatin, so it's relaxed, it's active, it's producing messenger RNA, so it's not really visible. However, there is always a structure, one or more structures inside the nucleus. So imagine this picture is a electron microscope picture, which shows you the whole nucleus. This is the whole nucleus here. Inside of cells that are not dividing, there is always one or more of these dark areas referred to as the nucleolus, if it's one, or nucleoli, if it's more than one. And these are active areas where the ribosomal RNA is made and is combined to form the ribosome, or the ribosomal subunits. They're still separate at that time. So these units that later on, we'll see when we talk about translation, are used to convert the information on messenger RNA into proteins. So the RNA, these are made by RNA and proteins. The RNA, or ribosomal RNA, that makes up this large subunit is produced inside the nucleolus or nucleoli if there are more than one. This structure disappears when the cells enter the cell cycle to divide. So you see at the end of the interphase, what is called, these structures before the cell enters what is called mitosis. We'll talk about that in the second half of the semester. When the cell enters mitosis, these structures disappear. And instead, the DNA, which is now relaxed, they start forming those visible structures, which are the chromosomes. Second component of the endomembrane system is the endoplasmic reticulum, or ER. Some people include also the nuclear membrane inside this part, the rough ER, one of those two components. Now, no matter what type, there are two types morphologically defined. One is called the rough. It's called rough ER because its appearance, it's rough. Under the electron microscope, you see all these dark dots corresponding to ribosomes, which are attached to it. The other one is referred to as the smooth ER, and unlike the rough ER, doesn't have ribosomes, so under the electron microscope, it looks like a smooth area. Not only that, but also structurally, they look different also in terms of the organization of the vesicles. So both of them are made by membrane channels and vesicles referred to as cisternae. So, but in the rough ER, they look like sacs, flat sacs, whereas in the smooth ER, these cisternae looks like pipes, basically. If you cut them, they look like circle pipes here, which are interconnected with each other through membrane channels. You can see some here. This will be a channel connecting these sacs. And now, they have different functions. Okay, we'll discuss them in the next slide. And so the relative amount of rough ER or smooth ER in cells changes. So you may have more rough ER than smooth ER in some cells, or vice versa, depending on the function of the cell. So first type is the rough ER. Again, some people include the nuclear membrane to the rough ER because the nuclear membrane, if you remember, it has two things. It has a double membrane with ribosome attached to it, exactly like the rough ER. Not only that, but it's also in continuity. There are some membrane channels connecting the nuclear membrane to the rough ER. So what is the function of the rough ER? It's related to the presence of ribosome. So what happens in the rough ER? There are proteins that are translated, or mRNA that are translated into proteins in these ribosomes. They are anchored here. And as soon as they're formed, these mRNA, they either get released or they enter the rough ER. Those that enter the rough ER, they enter into the rough ER because there they can be modified by adding usually sequences, for example, of carbohydrates, forming what we call glycoproteins. They're proteins modified with the presence of some carbohydrates. And then these proteins are modified here, and then they get transferred to the Golgi through the formation of vesicles. Like this one right here that is budding out. They're usually formed at the end of the cisterna here, the budding. So this vesicle is formed and then will separate and will fuse later on to the Golgi. The second type of endoplasmic reticulum is called the smooth endoplasmic reticulum. As we say before, under the electron microscope, like in this case here, there are no ribosomes, so it doesn't look as a rough surface, unlike the rough ER. You can see all these dots here that correspond to ribosomes. And also, they look, the cisterna, they look like basically like pipes joined together through membrane channels. Now, the function of the smooth ER is different than the rough ER. It's not related to protein synthesis and modification, but it's related mostly to lipid productions. So the lipids are there, and in some cells, not all the cells, but like for example, in your liver cells, the smooth ER also is important for detoxification. There is a lot of enzymes inside there that do degrade, for example, ethanol, and also they're important, basically in all cells, for calcium storage. The concentration of calcium inside these pipes here is 1,000 times higher than in the cytoplasm, because it's calcium is actually pumped in and released depending on the status of the cell. These smooth endoplasmic reticulum is usually referred to as sarcoplasmic reticulum when we talk about muscle cells. You see in BIO359, when we talk about those cells, we'll talk about sarcoplasmic reticulum. That's basically endoplasmic reticulum with a very specific function. Now, all the material that's gone through the endoplasmic reticulum, it doesn't get shipped directly to the cell membrane or other organelles. Usually it gets shipped to the next organelles or series of vesicles, if you want, or cisterna, sacs, which is referred to as the Golgi complex. Is referred to as the Golgi complex using the name of the scientist Camillo Golgi, who actually first described it by looking at cells under the microscope. So these organelles, again, receive the material that's gone through the smooth or rough ER through the production of vesicle by the rough ER and the smooth ER, such the one that I was showing you here, budding out. This vesicle will move and then will join the Golgi on a specific side, which is towards the ER, which is referred as the cis Golgi or cis phase Golgi. This is where these vesicles come in, they fuse with the membrane, and they release their content. So these could be proteins that be modified or lipids produced by the smooth ER. Once the content is released inside the Golgi, this finds itself in this series of plaques, 3 to 8 of them. They're not interconnected in this case, but they communicate with each other through vesicles. And there, you may have different things happening. One of them is some proteins get further modified by adding, for example, lipids. In this case, you're forming not glycoproteins, but lipoproteins. Or some other, molecules there also get basically packaged. Okay, so one of the functions of the Golgi is actually to properly direct the material inside to the different compartments. So this material that gets in here can end up, let's say, shipped to the mitochondria or the chloroplast or the cell membrane, or forming organelles such as the lysosomes, as we said before. And this is done when the material is shipped or lysosomes are formed. This is done by forming a new set of vesicles, which would have information in the form of proteins on their membrane to where they have to go and what they have to do. And these vesicles, again, here, they're formed by the opposite area of the Golgi refers to as the trans phase, and they will be shipped to either the cell membrane or the lysosome, or will form lysosomes or their organelles and so on. Now, one organelle that is formed from the Golgi through budding from the Golgi are lysosomes. These are digestive organelles, very important in eukaryotic cells. They have the function to basically digest anything that has to be digested inside the cell. What are these things? For example, imagine a cell like a macrophage. These are cells, first-line cells in the immune system that do ingest like viruses or bacteria too. And basically when they do that, they encapsulate this material inside a vesicle. So for example, a bacterium that was outside will get encapsulated inside membrane, which then finds itself inside the cell, the macrophage. The bacteria obviously can't stay there inside the cell, has to be digested. And this is done through fusion of those membrane, these vesicles that are formed with the bacteria inside, with these organelles, lysosomes. How do they work? Well, they work because they have tens of different enzymes inside here, which are able to degrade pretty much any form of organic molecule, such as protein, carbohydrates. So they have a bunch of hydrolytic enzymes there. Now, because of that, and this vesicle can be used also to digest, for example, old organelles, mitochondria. They get recycled all the time, the material mitochondria. They degenerate very quickly. They have to be reproduced all the time. And so the all mitochondria, they get digested exactly by lysosome fusing to the all mitochondria. Now, obviously, because these vesicles, these lysosomes, they have tens of very powerful hydrolytic enzymes, they can be extremely dangerous for the cell itself. Imagine one of these lysosomes breaking up, lysing on its own. You would have all these enzymes being released in the cytoplasm. And obviously, these enzymes would degrade anything they find in the cytoplasm too, such as ribosomes, the proteins, the cytoskeleton, all the organelles. They would start basically killing the cell. How is this prevented? Is by, or evolution has led to preventing this by basically selecting for enzymes inside the lysosomes that are very sensitive to pH. So they work very well in an acidic pH, like 5, but they don't work well at a pH higher than that, such as 7, and which is similar to pH inside the cells around 7.2. So inside the cytoplasm is around 7.2. So if any of these lysosomes ever burst, these enzymes will be released in the cytoplasm, they would find themselves in a pH of around 7.2, which is not compatible with their activity. So they would actually get turned off. That's a good way for the cell to protect itself from these organelles or the malfunctioning of some of these organelles. Other organelle that you find in eukaryotic cells are mitochondria. We'll talk about them in more details when we talk about cellular respiration. But as you know, these are important organelles to basically do the oxidative part of cellular respiration. Not the entire cellular respiration, but only the oxidative part. And so structurally speaking, they're made by two membranes. If you remember, this is one of the two organelles that produce it, or that has a lot of properties compatible with the endosymbiotic theory, explaining the how organelles were formed in eukaryotic cells. So mitochondria, they have two membranes. They have an outer membrane, which is a flat membrane right here, is smooth, and is fairly leaky, is not very selective. Some people refer to as a vestigial membrane, like some membrane that is left behind, compatibly with the endosymbiotic theory. Together with that, they have also another membrane here inside, which is referred to as the inner membrane, which folds inside, forming this structure called cystae. Cystae are those structures where the electron transfer chain in cellular respiration is performed, and also where the ATPase that structure that converts basically a proton gradient into ATP, leads to the production of ATP. They're all located here in the cyste. And the function of this folding is also exactly to increase the surface so that you can have more electron transfer happening, more ATP is working, more ATP formed. Now, this defines also an internal compartment, which is referred to as the matrix, where there is a circular DNA, there are ribosomes, and enzymes that do actually perform also transcription and translation of some of the genes in the DNA itself. Finally, last structure we talk about all eukaryotic cells is the cytoskeleton. So the cytoskeleton is a structure made by several proteins forming different type of fibers or filaments, which have different properties and different function. So now, the function, overall function of the cytoskeleton are different. They're there to support the organelles. We always imagine these organelles as free-floating inside the cytoplasm. In reality, they are anchored to the cytoskeleton. They move because they're anchored to the cytoskeleton by walking through the cytoskeleton. Some of these component cytoskeleton, the microtubules, you see when we talk about cell division, mitosis and meiosis, they form what is called a mitotic spindle, which are this structure, still part of the cytoskeleton, that help the chromosome walk towards opposite when they have to be separated. So there are three structures that are part of the cytoskeleton. You have microtubules, again, they're larger ones. They're around 25 nanometers. These are also the component of the flagella. You see the next slide. And then you have intermediate filaments. They are called intermediate because size-wise, they're intermediate between the microtubules and the next set, which is the microfilaments, as the name says. Micro means small, which are made by this actin. All of these structure here, the cytoskeleton is a very dynamic structure. You can actually look under a fluorescent microscope and you can see how it keeps changing. You can actually visualize some of these proteins with some fluorescent antibodies or molecules, and you can see how they keep changing. If you look a certain moment, a few seconds later, this structure can actually may have been reorganized. You see when we talk about microtubules, how they're structured is lengthened or shortened through depolarization or polarization of these tubulin proteins. Last structure is, and it's in continuity with the cytoskeleton. There are these structures that are structurally related, but they are responsible for the movement in different organism movement, both of the cells or the materials of the cells, which are referred to as flagella and cilia. Flagella are longer structures. They're made by microtubules, same component of the cytoskeleton, which then can move, and it allows it to move kind of in a wavy way. For example, these are sperm cell, they're very long flagella, which are responsible for their movement. This movement is not only like wavy, but is also circular itself. And then you have other structure which are similar to flagella, structurally speaking, but they're referred to as cilia. Cilia often form, they are encapsulated by membrane, and they often form this structure that can move basically like waves. Imagine like if you're looking underwater, you look at algae moving around, similar to that. And oftentimes these are used to move material. For example, your lung cells, they are part of your lung cells, have cilia, which are used to move the material in one direction, which is what forms the mucus that then you usually remove from your body. I won't say anything bad words, but the mucus that is then used basically is moved towards the exit, all right, the upper part of your respiratory pathway. And by doing that, by being moved in that direction, is also move anything that gets trapped on it, including like dust or small viruses, any particle that enters your airways can be removed through the movement of the mucus mediated by cilia. Finally, last thing that we're talking about here is a few structures which are unique of plant cells. We talked about eukaryotic cells. Plant cells are eukaryotic cells. So they have all the structure that we talked about until now, but they have also some unique structure which are found only in this type of cell. First one is chloroplast. You perfectly know what chloroplasts are these organelles that have the function to perform photosynthesis. So to capture the energy, the photons coming from the sun, and using them to, or storing that energy in the form of organic molecules, such as ultimately glucose or other molecules. So structurally speaking, like mitochondria, they're made by two main membranes. So there is an outer membrane and an inner membrane, but they have also a third system of membrane here, which form these sacs called thylakoids. These are the sacs that are super rich in pigments, and therefore they have the function of capturing the light. And that's what really gives the green color, because of chlorophyll, to the plant cells that do contain them. And together with that, they have an inner space here, which is not referred to as the matrix, but is referred to as the stroma. We'll talk about the chloroplasts and their structure as well in more detail when we talk about photosynthesis. Another important structure that is found only in plants and not in other or in animal cells and so on, plant cells, is the vacuole. This is a very large structure that's which is found in the inner part or towards the central part of the cells, hence the name central vacuole, which has different function. It's extremely large. It can take up to 90% of the cell volume, so it can be really, really big. It's very liquid, rich in liquid, and it can be used to store inside salts, organic molecules such as glucose and so on, acids and so on. Lots of things can be stored in there. Together with that, it can be used also for two purposes. The water inside here and the component inside creates basically a pressure going outward from this structure, which basically means it's pushing the rest of the cell outward. This is the cell rigidity. Okay, it gives support to the cell structure itself, compensate with the pressure outside. And so it's used for rigidity and also for cell growth. Some cells do grow just by expanding the central vacuole. By doing that, they actually get longer and longer or larger and larger over time, just by expanding the central vacuole there. And forgot to tell you, the membrane surrounding the form central vacuole is referred to as the tonoplast. Last thing, structure for plants is the cell wall. Not all eukaryotic cells they have not all eukaryotic cells have a cell wall, but plant cells that do have, all cells would have it, which is a structure that gives rigidity to the cell. All cell walls, they do contain cellulose. You remember that polysaccharides formed by beta glucose. Cellulose fibers joined together by hydrogen bonds make the cell wall extremely strong and rigid. Some other cell walls, so those containing cellulose only, are referred to as primary cell walls. Some others do contain about 30% of cell types in plants do contain also a second component, which is lignin, is a polymer of different molecules, which makes the cell wall even stronger. You see the cells when we talk about plants in BIO359. The cells, for example, are responsible for the movement of material through the plants do contain lignin, making it very strong and harder to collapse. So they help also protecting obviously the cells from anything from outside, such as bacteria entering. Because of that, the presence of the cell wall represents a barrier also to movement of material in and out, and also between cells. So oftentimes cells which have these cell walls that are connected, so there are some opening within the cell wall which connect the cytoplasm of neighbor cells, like in this case you can see right here. So this will be one cell, This is another cell, and the cell wall will be here. You have this structure referred to as plasmodesmata that do allow for cells to communicate with each other and exchange material. So they overcome that barrier that is the cell wall represent in plant cells. Finally, summarizing everything that we talked about in this lecture. So we went through a fairly quick and not super detailed overview of cell structure. So first of all, we talked from a historical point of view, how we got to the discovery of the cell and the function of the cell. Then we talked about the shared basic features that all living organisms, so prokaryotes or eukaryotes, they all have. The cell membrane, area dedicated to the DNA, and the presence of a cytosol. And then we went into the specific structure present only in prokaryotic cells, eukaryotic cells, and then we finish up talking about plant cells.